Thanks for inviting me to share my research so far. And obviously, this is a work that's funded by Cure Cancer. I'm from the University of Adelaide, but I'm also affiliated with the Basel Hetzel Institute, which is the essentially research institute of the Queen Elizabeth Hospital, which I'm showing here. So the research I'm working on is like trying to find treatments for metastatic bowel cancer. So metastatic bowel cancer is the second leading cause of cancer-related death worldwide and even in Australia. About half of the patients, really the reason why bowel cancer is so deadly is because these cancers metastasize. Now, as part of the Queen Elizabeth Hospital, I'm working with the Department of Surgery, Upper GI. So we work a lot with the surgery team, where we try to improve the treatment outcomes for these patients. So the individual survival rate for metastatic bowel cancer is 15%. There's a very small proportion of these patients that are very lucky in that they will undergo what we call curative liver resection. So just for looking at the basic biology of where the colorectal cancer or colon cancer is, liver is the main site of metastasis for these patients. So even though we call it curative resection, the 10-year survival rate for this bowel cancer is still 25%. So it's really critical to find new treatments for these patients. Now, there's a lot of talk about immunotherapy so far. Actually, I need to move my head or something because like the sample I'm looking at is not in my image. But anyway, there's a lot of different immunotherapies for that are currently available, right? And a lot of the talks that we've heard so far are on immune checkpoint inhibitors. But that's not the only type of immunotherapy available. I'm sure you guys are all aware of that. So I'm not going to talk about most of them, but I'm just going to focus on this cell subset, which you can see here if I move my head around, which is called cytokine-induced killer cell therapy. So in this therapy, it's I'm just going to shift to the next slide so you guys can see it better. So it's not a new therapy. It's quite old, actually. The first description of it was back in 1990s, where if you collect PBMCs, so peripheral blood cells from patients, and you differentiate them in a certain way, what you end up generating is this population of NK-like T cells, where they're primarily T cells that express particularly as shown here. CD56, which is a classical marker to identify these cells, but also the natural killer cell receptor. So they can, in theory, detect cancer cells both in MHC-restricted and unrestricted manners. And once they recognize the tumors, they're like your other killer T cells. They can actually function by killing the cancer cells through their cytolytic activity, but also by promoting a type 1 response by secretion of interferon gamma. So we were really interested in this therapy because of a couple of things. One, we're not a big laboratory here at the Adelaide Uni, but at the BHI. So we really wanted something that we could achieve and provide to these patients eventually. And the advantage of SIC therapy is that the production methods are quite achievable. So there's a very low barrier in terms of the cytokines and activating materials that you actually need for it. So other than being able to produce it quite easily, the cost is not that high either. And like I said before, because there's TCR dependent and independent cancer recognition, it's much more available for a lot of patients. So for example, in colorectal cancer, actually a very, very rare small proportion of patients, which is the microsatellite unstable patient, benefit from checkpoint inhibitor blockade, but they're only like around less than 10%. We think that we can provide this therapy to all the patients that come through the hospital if we do actually implement it. So the first step that we've done, so we published this in 2023 by my PhD student that I was co-supervising, who's finished now, Celine, was we just wanted to collate all the evidence on CIK cell therapy. And so we published this paper in JITC, which is a systematic review, looking at really the clinical benefit of using SIK cell therapy for colorectal cancer. Now, this review article, you're free to have a look at it. It's very clinical, but I'm more of like a visual person. So here's just like a survival graph instead that you can actually see and appreciate from one study, where you can see here those patients that receive SIK cell therapy in red. So this is a retrospective study, so it's not a specific clinical trial, where these patients have a very significant extension of their overall and progression-free survival. And this was a retrospective study from stage 1 to 4. And recently there was this other study that published this result purely on untreated metastatic colorectal cancer patients. And again, as you can see, the red here is those patients that are receiving immunotherapy. you can see extension of survival over 5 years, where almost 70% of these patients are still surviving with good progression-free survival as well. So we see this really strong clinical benefit, and this is really what was reflected in our systematic review. So where we compared essentially in the studies that had comparison groups of those that received SIG therapy or standard therapy as control. Most patients that have received SIG therapy had improved overall progression-free survival. And what's important here for us was there was very low or limited reports of toxicity induced by this therapy. So this gave us the confidence to potentially really want to push forward in trying to deliver this therapy here in Australia, and starting with obviously in Adelaide. So basically, we want to develop this program based on the clinical findings that we've seen before, where we can potentially provide SIG cell therapy as an adjunct therapy, so in combination with standard treatment for these patients here. And that's really the main goal of my research that's partly funded by Cure Cancer, which is two main goals. First is establish the clinical framework to actually deliver this therapy to the patients that we have here. And the second thing really that might be much more interesting is to identify fundamental knowledge gaps in CIK cell biology so that potentially we can develop and improve this therapy further. So I'm just going to focus first a little bit on the first bit, and these are all published papers, so I'm just going to fly through it, hopefully. So the first thing that we wanted to do was we wanted to, as I said before, establish the clinical framework. So how we approached this was based from those clinical trials that was published. Could we find a unifying pattern that we can use to actually adopt and develop in Australia, so we're not starting from scratch? And we've done that. We've summarized that literature here that was published last year in Cancer Treatment Reviews. And this is really just our general framework, which is not going to be very different from a lot of people who are working with adoptive cell therapy. So we plan to get some blood from patient, and if we can get some biopsy as well, generate tumor organoids, expand the SIG cells, validate that they are effective, and then reinfuse it to these patients. Now we haven't only done this theoretical framework, but we've also actually started to do some validation that we have capacity to generate these SIG cells. So this was published as well last year in Cancer Immunology and Immunotherapy, where we generated SIG cells from liver metastatic colorectal cancer patients that have undergone resection or surgery here at the Queen Elizabeth Hospital. And I'm not going to show the data for this, but just to summarize it, we have essentially developed a production protocol that should be clinically appropriate so we can deliver this therapy for these patients. We've validated that our SIG cells are actually having similar molecular features to what's published out there. And I think what's important here is we had a broad range of different types of patients that we had tried to generate SIG cells from. And what we can say is it seems to be like age, sex, or chemotherapy exposure. So some of these patients will have undergone chemotherapy before we can get blood from them. It didn't really affect how well we can generate the SIG cells from them. So these cells that we generated have the same anti-cancer activity that we can see using both different ways of assays. So basically, we've used cancer cell lines grown in 2D and 3D, and also using patient-derived tumor organoids from the surgical resection samples that we have. And I'm going to try to focus it a little bit more later on this is what the key points that we think where our fundamental research can potentially improve this therapy on is the variability in CIK cell production and cytotoxic activity as well. And nevertheless, we think that as it stands, we think that this therapy already should be potentially will benefit our patients. So really this year with our oncology team here, we've developed this clinical trial proposal, and we're actively seeking funding for it. We're calling it the IMPACT CRC trial, which is implementation of CIK cell therapy for colorectal cancer treatment. And I'm just going to give you guys a brief overview of what the trial is going to look like, which is shown here. So what we plan to do here is we're going to recruit treatment-naive metastatic colorectal cancer patients, because we think that these are the patients that will likely benefit the most from this therapy. And the trial intervention that we plan to do is provide 4 doses of SIG cell therapy on these patients, provided once a month as shown here. So after 4 months, we're going to follow them up for 2 more months, not really to look at survival because it's a pilot study. Eventually, we do want to see that, but really just to see if we can develop the clinical production framework here locally in Adelaide to produce these cells and then to deliver it to our patients. So just to give you a microscopic view of actually how we should generate these cells, this is our diagram for this. So basically, our plan here in terms of why it's quite neat, I think, is like we don't need to get a lot of blood from these patients. Our dosing is actually about quite high. We want to inject 5 billion cells per dose for these patients, but we should be able to generate that amount of cells from 50 mils of blood from the PBMCs from these patients. So the idea here is like for the 4 doses, we're going to collect blood each time and produce them freshly. As I said before, give them 4 doses. We want to culture our cells for 14 days, and then there's going to be 2 weeks to validate the efficacy and also sterility of this before we reinfuse them back into our patients. So as I mentioned before, our key outcome measures really here isn't really to see clinical outcomes just yet. That's for bigger trials later on, but really our funding proposal or what we're asking for in the future is like to monitor our ability to expand 6 cells and to develop a clinical pathway, a practical clinical pathway where we generate in the lab and deliver it to the patients at the Queen Elizabeth Hospital initially and potentially at the Royal Adelaide as well, which are the 2 major hospitals at South Australia. So the second aim, like I said before, is the fundamental knowledge gaps in CIK cell biology. And this is what you normally get in terms of when you do a flow cytometry to validate your CX cells. So as you can see here, the double positives, this is at day 21. When you look at a PBMC, this type of gating, you're going to get around 1 to 2%. So we are able to generate about, at day 21, around 30 to maybe 40 to 50% of our populations are double positive. At day 14, it's around 20%. However, one of the things that really, when I was looking at the literature of the biology of these cells, is like the understanding really of what the double positive cells are, what they are actually is very limited. And in fact, if you just do like a very, very basic flow cytometry plot, so this is not a single cell sequencing data, but I'm going to show some of that later on, where you just pick a few markers here and there that's really relevant for T cells, cytotoxic T cells. You can see here that distinct clustering. And as I said before, I've really highlighted the cells, the double positive cells in blue here. At day 0, or basically at the PBMC stage, they're very rare, and you expand them over time. But you can see this clustering or heterogeneity present in our cell therapy. So really, it spurred on to the question is like, what are the critical cellular components in CIK cell therapy? And the idea behind is like, if we find that there's actually a more important subset present in this therapy, maybe we should find ways to improve the expansion of these cells further and less on like making it a very heterogeneous process. So how do you actually generate CIK cells? So it's a very simple process, as I said before, and that's what made it attractive for us. So you harvest PBMCs from patients, you do a FICO spin to get, like I said, the PBMC. You prime them with interferon gamma for 1 day, and that apparently makes a lot of like imprinting changes or epigenetic changes to these cells. Then you do standard, very simple anti-CD3 stimulation with IL-2, and you just refresh IL-2 every 3 to 4 days, and the cells are ready by between day 14 and 21. So when I started this project, a lot of new papers are coming out on making single cell atlases, and I thought, oh, that's perfect. We should make a single cell atlas for CIK cells, and hopefully we can figure out what these populations are. So for the experiment I'm going to present to you today, it came from 5 treatment-naive metastatic colorectal cancer donors, cultured using our GMP-compliant protocols. So then we're not really going back and forth in terms of the clinical protocols. And we analyzed day 14, and thinking that potentially there might be some intermediary cell types there that we can capture in our single cell data set. And just to show you like our flow cytometry validation plot for what our cell culture products look like at day 14, you can see here that, like I said before, it goes from like around 1 to 2% in your PBMC to about 18 to 20% on average from our 5 donors. So this is our single cell sequencing data. I'm not a bioinformatician, but lucky enough, I was able to recruit a PhD student who took on the task of becoming a bioinformatician. And you know, as if you've actually analyzed single cell RNA sequencing data, especially for cell types where you don't really know or you don't really have an expectation of what you see, one of the hardest things that you can do is like try to understand how many clusters are actually present in our data set. But I think we've settled on that we think that there are 19 distinct clusters present in this culture. So it's still a work in progress, but we are sticking with 19 at the moment. And here are the distribution of our of our 19 clusters across the 5 donors. And what you can hopefully see, if I move my head around, is that it's very consistent, except for this one patient, which we had a little bit more of cluster 0. But it looks like from the rest of the clusters, disproportion of these cells, when you culture it this way, consistently generates these 19 clusters in very similar proportions. So basically,
Kevin, just 1 minute to go.
Yep. Okay. So I'll finish this really quickly. So all I'm going to say here is like the 6 cells we've generated look very similar via functional profiling. We identified 7 potential cytotoxic clusters from this, and basically using manual curation of cytotoxic genes here, combining them into the cytotoxic score, we can show that the 7 clusters had the highest score for it. We also were able to identify our progenitor clusters as shown here using this program called CytoTrace, which allows us to score our cell clusters based on their differentiation status. And using that information, we used our progenitor clusters as our starting point for molecular differentiation pathway analysis. So this is going to be my final data slide. We think that we've annotated them. Like I said before, this is still preliminary data, but just to show some interesting things here, you get CD4 cells, CD8 cells. But here are some things that I thought was quite interesting, where we've also generated cytotoxic CD4 T cells. There was actually a population of Th2 regulatory T cells, and also a very small cluster of gamma delta T cells, which was never presented before. So our next steps for this project is, like I said before, we are pretty much like ready for doing a clinical trial. We just need funding for it. We've optimized our bioreactor-based production profile for this, and we're just applying for our human ethics application now. For our fundamental studies, we are going to validate our CI clusters using flow cytometry just to show that the protein works. We're going to obviously assess the tumor killing capacity of our 7 known cytotoxic clusters, and really develop a differentiation atlas of the CIK cells. So we actually have generated single cell data as well, not just on day 14, but following from PBMC, day 7, 14, and 21. So that's under analysis at the moment. So we've just sequenced them. So yeah, so I'd like to thank our main group here. So basically, it's really through clinical leaders such as Professor Guy Maddern and Tim Price, who's an oncologist and surgeon, who's really vital for, I guess, like patient sample collection, and allows us to really try to push this further to the clinic and not just as a research study. And also my PhD student, who's now a postdoc, Celine Lee, Yoko, who's our medical oncologist, who's leading the project, and Elham, who's a PhD student, who's analyzing single cell data. So thank you for listening, and I'm happy to take questions.